Dear my students, we will talk today about Introduction to Pharmacology. In this lecture, we're going to learn some terms very important to understand now. If we understood these terms, then whatever we will take in the future will be easy to be understood. So let's go first to the Now, pharmacology can be defined as the study of a substance that interacts with living systems through chemical processes, especially by binding to regulatory molecules and activating or inhibiting normal body processes. نخلي نسهل الموضوع إنه هو فد مادة كيميائية تقوم بأنه ترتبط ويصير ارتباط مباشر بجسم حي من خلال مكان منظم أو نسميه مستقبل أو ريسبتر. هذا يؤدي بعد الارتباط إلى أما تحفيز أو إيقاف تحفيز فعالية من الفعاليات، وهكذا تكون فكرة الدواء أو أي مركب كيميائي. هذه المواد أما chemical administered to achieve a beneficial therapeutic effect on some processes within the patient, or chemical has toxic effect on regulatory processes and parasite infecting the patients. يعني ممكن أنه ننطيه كعلاج للباراسايت. Here we have an important term called medical pharmacology. Sometimes, is there any differences between pharmacology and medical pharmacology? Yes, definitely. Because in medical pharmacology is often defined as the science of substances used to prevent, to diagnose, treat disease. What does these three terms mean? What is the meaning of prevent? It means we have a drug, when we pass it to the patients, we got a prophylaxis from a disease. Like for example, عندنا مثلا patients got asthma and his lungs sensitized from cold weather, and tomorrow there is a news saying that tomorrow there is a cold weather. What you will do you wait until the cold weather comes and then the patients got the episode of asthma? No, definitely. What we will do, we give a drug to the patient, it prevents the episode of asthma even the cold weather is come. Okay? Diagnose, that means we give a drug to give a differential diagnosis from a disease such as in Cushing syndrome. Treat, that means the treatment. This is definitely. Now, the other term that we should learn is the toxicology, which is a branch of pharmacology that deal with the undesirable effect of chemicals on living system. That means the mechanism of these substances that give the undesirable effect. Pharmacogenomic. This term is very important actually. The relation of the individual genetic makeup to his or her response to specific decoding of the genome of many species, from bacteria to human, has led to the recognition of unsuspected relationship between receptor family and the way that the receptor protein have evolved. طبعا اكو big revolution nowadays trying to design the treatment in accordance to the genome, للخريطة الجينية للمريض. بمعنى أنه مريض ممكن إذا أعطي دواء يستقبله بطريقة غيره لا يمكن أن يستقبله بهذه الطريقة، بمعنى أنه الفعالية الدوائية عند فلان تكون أعلى من الفعالية الدوائية عند الآخر، فلذلك أحيانا نحتاج أنه نسوي ديزاين لكل مريض، وهذا فد big revolution. Similarly, short nucleotide chain called Antisense oligonucleotides synthesized to be complementary to natural RNA or DNA can interfere with the readout of gene and the transcription of RNA. These intracellular targets may provide the next major wave of advance in therapeutic. Hopefully, we hope. Well, student, look to this. What is the nature of a drug? The drug molecule interacts as an either agonist, that means it leads to activate, or antagonist, that means it leads to inhibit with a specific molecule in the biologic system that play a regulatory role. C. Chemical antagonisms may interact directly with other drugs. Drugs may be synthesized with the body, such as hormone, chemical not synthesized in the body. Here, poisons are drugs that have almost exclusive harmful effect, while toxins are usually defined as a poison of biologic origin, that's to say synthesized by the plant or animals. That means when you hear this is toxin, it means it is synthesized by plant or extracted from plant or animal, in contrast to inorganic poisons such as lead and arsenic. This is also very important. Well, let me explain this clearly. What does chemical or chemical antagonist? That mean, for example, patient عنده خلينا نقول تسمم ب اسيتيل ساليسيليك اسيد. تمام. شلون انطي ادراج يعاكس هذا الفعل اي يعاكس الاسيتيل ساليسيليك اسيد؟ كمثال انطي ديفينتلي صوديوم بايكربونات. ليش؟ لان اذا اجت كقاعده ويا اسيد ايش راح يصير؟ راح يصير ايونايزيشن. لو ان ايونايزيشن راح يكون هناك ايونايزيشن، فبالتالي ما يصير شنو؟ ري ابزوربشن. اذا كان هناك ايونايزيشن ما يصير ري ابزوربشن ويصير get rid from the excess amount of acetyl salicylic acid. هذا شو نسميه؟ نسميه chemical antagonism. Well, the other thing here, we will talk about the drug molecule. Every drug molecule must have the appropriate size, electrical charge, shape, atomic composition to interact chemically with its receptor. Therefore, a useful drug must have the necessary properties to be transported from its site of administration to its site of action. Finally, practical drug should be inactivated or excreted from the body at a reasonable rate so that its action will be of appropriate duration. The physical nature of drugs. Drugs may be solid at room temperature, such as aspirin, atropine, liquid, such as nicotine, ethanol, gaseous, such as nitric oxide. These factors often determine the best route of administration. 4. Organic compounds, such as carbohydrate, protein, lipids, and their constituents are all represented in pharmacology. 5. Oligonucleotides in the form of small segments of RNA have entered clinical trials and are on the threshold of introduction to the therapeutic, while the last one is the inorganic elements such as lithium, irons, and heavy metals. All these is actually the physical nature of drugs. Now, if I will ask you what is the physical nature of drugs, simply you will say solid, liquid, gaseous, organic, inorganic, and oligonucleotides. طلاب، بالإضافة للي حكيناه سابقا many organic drugs are weak acid or weak base. حقيقة هذه لها فائدة كبيرة إنه نحدد الدواء مالتنا هو weak acid أو weak base. يفيدنا بالsolubility، يفيدنا بالabsorption، يفيدنا بالvolume of distribution، بالايونايزيشن، بالكاينتيك مال الدواء. بحركية الدواء من لحظة الادمنستريشن إلى أن يصل إلى المكان اللي المفروض يوصل له. فلذلك النيتشر of أو طبيعة الأورجانيك drug whether it is weak acid or base. Well, drug size. The molecular size of a drug varies from very small, such as lithium, molecular weight 7, to very large, alteplase, with a portion of molecular weight of 59,050. However, most drugs have molecular weight between 100 to 1000. The lower limit of this narrow range is probably set by requirements of specificity of action. To have a good fit to only one type of receptor, a drug molecule must be sufficiently unique in shape, charge, and their properties to prevent its binding to other receptor. Now the point is to achieve such selective binding. It appears that a molecule should in most cases be at least of 100 molecular weight unit in size. The upper limit in molecular weight is determined primarily by the requirement that the drug must be able to move within the body from the site of administration to the site of action. Drugs much larger than molecular weight of 1000 do not diffuse readily between compartments of the body. Therefore, very large drugs, usually protein, must often be administered directly into the compartment where they have their effect. In the case of alteplase, for example, which is clot dissolving، يعني هذا نطيب حالات الجلطة بحالات الجلطة اللي يصير بالقلب الأوعية الدموية، ننطيه مباشر. The drug is administered directly into the vascular compartment by intravenous or intra-arterial infusion. Why? Because it is very large, more than 1000 molecular weight. Well, this slide will talk about the drug reactivity and drug receptor bond. Actually, it is very interesting. Drug interact with the receptor by means of chemical forces or bonds. There are of three major types: covalent, electrostatic, and hydrophobic. Well, let's go through this. Indeed, covalent bonds are very strong and in many cases not reversible under biologic conditions. Thus, the covalent bond formed between the acetyl group of acetyl salicylic acid and cyclooxygenase, its enzyme targeted platelet, is not readily broken. As it is not broken, the platelet aggregation blocking effect of aspirin lasts long after free acetyl salicylic acid has disappeared from the bloodstream. Do you know why, student? Do you know why? Well, I will tell you this. Because this covalent bond, the strong covalent bond, leads to conformational change in the receptor. That means the receptor no longer the previous one, and the body needs to synthesize a new one, a new receptor. That's why the effect of aspirin lasts long time and is reversed only by the synthesis of a new enzyme in a new platelet process that takes several days. Other example of highly reactive covalent bond forming drug include the DNA alkylating agent. حقيقة هذا الجروب من الالكيليتنج agent يستعمل بالكيموثيرابي بالكيميائي to disturb cell division in the tumor. In the electrostatic bonding, the other type rather than the other type here is much more common than covalent bonding in drug receptor interaction. Electrostatic bonding bonds vary from relatively strong linkage between permanent charge ionic molecules, hydrogen bond and very weak induced dipole interactions such as van der Waals force and similar phenomena. هذا هذا حقيقة جدا مهم طلاب. ليش؟ لأنه الهيدروفوبيك bond are usually quite weak and are probably important in the interaction of highly lipid-soluble drug with the lipid of cell membrane and perhaps in the interaction of drug with the internal walls of receptor pockets. The specific nature The specific nature of a particular drug receptor bonds of less practical importance than the fact that drugs that bind through weak bond to their receptor are generally more selective than the drugs that bind to mean of very strong bond. This is because weak bonds require very precise fit of the drug to its receptor. بمعنى بما انه هو البوند مالته ضعيفة فلو هو لو مو كلش سبيسيفيك إله ما كان من الممكن أن يصير البايندينج، لكن كون انه هو جدا سبيسيفيك هنا راح يصير شنو عندنا؟ راح يصير عندنا البايندينج. Well, if we wish to design a highly selective short-acting drugs for a particular receptor, we would avoid highly reactive molecules that form covalent bonds and instead choose the molecules that form weak bonds. A few substances that are almost completely inert in the chemical sense nevertheless have significant pharmacologic effect. For example, xenon, our inert gas, has an anesthetic effect at elevated pressure. Now, the shape of a drug molecule must be such as to permit binding to its receptor site via the bond just described. Optimally, the drug shape is complementary to that of the receptor site in the same way that a key is complementary to a lock. يعني هذه هي نظرية القفل والمفتاح، بمعنى أن الدواء يكون بالضبط الشيب مالته يقعد على الشيب مال الريسبتر. Furthermore, the phenomena of chirality stereoisomerism، هاي النظرية اللي أخذتوها بالمرحلة الثانية شباب، أرجو أن ترجعون للstereoisomerism، is so common in biology that more than half of all useful drugs are chiral molecules, that is, they can exist as enantiomeric pairs. Drugs with two asymmetric centers have four distereomers, such as ephedrine and some pathomimetic drugs. In most cases, one of these enantiomers is much more potent than its mirror image enantiomer, reflecting a better fit to the receptor molecule. وطلاب كثير من الأدوية يكون لها enantiomers أكثر من واحد، more than one chiral center، لكن تلقى واحد من عندهم فقط هو الأكتف، لذلك تحديده مهم. لذلك احنا من درسنا لكم بالعضوية في المرحلة الثانية حتى تفهم يعني الفانكشنال جروب شلون توزيعها بالفضاء، وهذا التوزيع يأثر على الفعالية الدوائية. And because of that, the more active enantiomer at one type of receptor site may not be more active at another receptor type. For example, type that may be responsible for some other effect. For example, carvedilol هذا alpha beta blocker. Carvedilol, a drug that interacts with adrenal receptor, has a single chiral centers and thus two enantiomers. Rational design of a drug implies the ability to predict the appropriate molecular structure of a drug on the basis of information about its biologic receptor. Until recently, no receptor was known in sufficient detail to permit such a drug design. Instead, drugs were developed through random testing of chemicals or modification of a drug already known to have some effect. هاي طريقة طبعا. A few drugs now in use were developed through molecular design based on knowledge of the three-dimensional structure of the receptor site. والحقيقة طلاب يعني عندنا احنا طريقتين بالراشنال دراج ديزاين حتى نصنع دواء. اما بطريقة التجربة انه مثلا دواء هو مسكن ألم، ونشيك على الضغط نكتشف انه عنده قدرة على تقليل الضغط، نشيك على السرطان نكتشف انه عنده قدرة على ايقاف نمو خلايا السرطان. هاي طريقة، ومن خلالها نقدر نعرف الريسبتر نشتغل عليه من خلال الفعالية الدوائية. الطريقة الاخرى اللي هي few drugs in now were used developed through molecular design، يعني تعرف الستركتشر ال 3 dimension structure of the receptor site، ومن عنده تحاول تقترح الفعل العلاجي. وكمبيوتر بروجرام are now available نسميه simulation that can interactively optimize drug structure to fit known receptor. يعني نسويها بالكمبيوتر ال 3 dimensional structure of the drug and the 3 dimensional structure of the receptor، ونشوف إذا يكون أكو بيناتهم binding أو لا، ومن خلالها نقدر نعرف لأي شيء ممكن يستعمل.